So hi there. Today we're going to be talking about skeletal muscle organization. When you're looking at a skeletal muscle like your bicep, you're looking at it from the outside and you can typically just see the bicep as a whole. But what you can't see is what's all on the inside. So when you look at skeletal muscle, it's organized as bunches of bunches, which means we have a smaller structure that's been bundled together to form a larger bundle, which is bundled together to form an even larger bundle. So this whole cascading set of events, we need to be able to break down and really explain. So we're going to start off today with the smallest part of our bundles, which is going to be our proteins, and we're going to build it all the way up to a whole skeletal muscle and then break it all the way back down again. So that way you can understand the way that skeletal muscle is organized. Okay, so we're going to start off today with proteins. In your skeletal muscle, you have 3 different groups of proteins: regulatory proteins, structural proteins, and contractile proteins. The first 2 groups we're not going to include in this demonstration. Regulatory proteins are important for turning contraction on and off, but they are fairly small in regards to contractile proteins. So we're not going to include them in this demo. Structural proteins, once again, very important because they maintain the structure of the skeletal muscle, but not going to be in this demo. We're going to look solely at the contractile proteins. Contractile proteins being myosin in blue and actin in yellow. Myosin is going to be the thicker filament, which I can't really tell because it's just one pipe cleaner, but if I was to twist it all together, this is the thicker structure. This is going to be in the center of your sarcomere. Towards the outside of your sarcomere, you're going to find actin. Actin is the thin filament. Both of these are going to slide past each other to create movement. Okay? So in looking at organization, what we do is we take a bunch of myosin and actin, organized into sarcomeres, and we group them together, which you can see here. Right? I've got my myosin in blue, my actin in yellow. So what I've done is I've taken them and I've grouped them together so that you can see the several repeating units of sarcomeres here. Okay? These are the protein levels. So this is still within the skeletal muscle cell or fiber. So if I take several of these and put them together, what you see if I have bunches or bundles together organized into a larger bundle. Okay? And that bundle is known as a skeletal muscle cell or fiber. You can go back and forth between the 2 terms. A skeletal muscle cell is correct. A fiber is also correct. Because a skeletal muscle cell is very long, it is often called a fiber. So terminology-wise, both of those are okay. So here's your skeletal muscle cell. And you can see all of my bundles of actin and myosin on the inside. Also, when you look at it, you can see that it's covered by 2 different types of coverings here. The first one in pink is called the sarcolemma. The sarcolemma is the cell membrane. So this is actually the border between what's considered to be inside the cell and outside the cell. Okay. Over top of the sarcolemma, I have this one in bright green here, which we call the endomysium. The endomysium is connective tissue, which is going to cover my muscle cell. Okay. So here is one muscle cell, and you can see that already I have bunches inside of larger bunch. Okay. Now what happens is I take these skeletal muscle cells and I combine them all together with other ones, right? As you can see here, all together. And then I cover them with a coating called perimysium. And this forms an even larger bundle that we call a fascicle. Okay? So you can see my skeletal muscle cells inside covered by the perimysium to form a fascicle. Okay? Now, this fascicle is going to join with other fascicles, like so. Other fascicles are going to join together. And then we're going to cover all of this with another connective tissue that we call epimysium. And that together is going to form your bicep. So that is your organization of your skeletal muscle. Now, we constructed it, so we need to deconstruct it. So the first thing we're going to do is we're going to take off the epimysium. That epimysium connective tissue is what is going to not only surround the skeletal muscle, but also continue on to form the tendons that are going to link that muscle to a bone, so that when the muscle contracts, it pulls on the bone to create movement. So when we take away that epimysium, we can see our fascicles. Right? Each fascicle is surrounded by the perimysium. Okay? And if I take that perimysium away, I'm left with my skeletal muscle cells. Okay? If I look at one skeletal muscle cell, and I was to unravel the endomysium and the sarcolemma, I'm left with bunches of actin and myosin. And if I continue to unravel those bunches, I end back with my proteins. So I hope this was helpful. Thanks.